you get your Scantron, yay! So don't forget the fact that Jerome and Lexi also have office hours. Jerome has his tomorrow at 9 to 10. So if you need help, that's where you are. I just want to remind you, and I'm not chastising anybody or anything, but please follow rules, okay? And the rule is, I'm completely, I would love to help you. If you have questions, I would love to do it, but as I said on the first day of class, I have my office hours, Jerome has his office hours, Lexi has her office hours, and if you absolutely cannot make it show up because of class schedule only, not because you have other things to do like work, then make an appointment to come in and ask, and do it many days before, a week before. Please don't come to me when you see me walking down the hall and then say, hey, can I ask you a question? That's not, and I've been told by evaluation that I'm not approachable. Well, you know, you don't do that to people, right? If it's important enough for you to ask questions, make an appointment, come into my office, and I'll give you an hour. I'll give you 2 hours if you want, but I am making you follow the rules. You are going to be professionals, and you're going to find out that if you don't have rules and regulations, people will definitely inundate you, and then you don't have enough time in the day to do what you need to do. So please, if you need help, I'm very willing to help, but please follow the rules. That's it. And this doesn't apply to all of you. You know who you are. It only applies to a few of you, and please follow the rules, okay? And you're more than welcome to complain about me, about anything you want to later on during the student evaluation. It's okay. But for now, please follow the rule. Okay. So this is roughly where we are. Exons. What's the function? Roughly the function of exons, again, is to, the exons are either independently or as a group, will work toward producing a domain for the protein, okay? And then here's the example of domains. And we're done with regulation of translation, okay? Transcription, as well as modification, okay? So a little bit left over is, this is actually, believe it or not, is a type of regulation for translation. Translation in general is degradation of RNA is actually a type of translation, translational regulation. How is it a type of regulation? When your body wants to carry out translation, it needs to know that the mRNA is properly made, synthesized, as well as properly processed. If an mRNA is not properly synthesized and not properly processed, guess what? It needs to be destroyed. And that's the moral of this slide, is that an mRNA, again, an mRNA that is not properly synthesized or modified, then it's destroyed. What would be one example? No cap, no poly A tail, still has the intron bound. Right? Still has spliceosome bound. Okay, those are examples of RNAs that needs to be destroyed. How about RNAs? Remember, RNA synthesis is completely error prone. So if the synthesis, if the RNA, if there's an indication that this RNA, there are mutations in this RNA, then again it needs to be destroyed. RNAs are destroyed in both the nucleus as well as the cytosol. So in the nucleus, there's regulation, right? There are markers of whether or not this RNA is a good RNA. If it's a bad RNA, then the cell will destroy it by degrading it. And then when you go out into the cytosol, there's another set of proteins that can regulate, that can detect whether this mRNA is a good RNA or bad RNA. And then if it's bad, then it gets degraded as well. Okay? Yes, ma'am.
So there's like double check. Once in the nucleus.
Once in the nucleus and once in the cytosol, right? Right. Again, the whole idea is that you don't have proofreading and you don't have an RNA repair system. right? So the only thing that can be done is to get rid of bad RNAs, and there are a lot of them. Right? There are a lot of them. Also, unused RNA. So when we use the word nuclear RNA, it includes a lot. It includes any RNA really that's in the nucleus. And so some RNAs might be useful in some case, but then they're no longer useful. So to recycle the RNAs are also degraded. So again, the idea of this is that, the last two slides is that that you do have degradation of RNA in both the nucleus and the cytosol. Examples I just told you. If you don't have a cap, if you don't have poly A tail, or if it's a defective RNA in some way. Okay? That and that leads into translational regulation of translation. In that again, if it's a bad RNA, then translation won't occur. So that is indirectly a type of regulation of translation. Just like transcriptional modification, just forget about the co and post. Eventually, if I remember it outside of lecturing, I'll get rid of that term. So basically, translational modification. Okay, and there are a lot. There are a lot. We already talked about a few types of translational modification. What do you think some of those are? Acetylation. Methylation. Anything that changes the protein in any way is a type of modification. Those are true post-translational modification. So anyway, so we'll start out with talking about the regulation of the initiation of translation. We'll kind of gloss over elongation of translation, and then we'll talk about the termination of translation a little bit. But we'll concentrate mainly on the regulation of the initiation of translation, and then skip over to translational modification. Okay, so regulation of translation. All of this is important. I'll go into each one of them in details, but basically for now, memorize this. Okay, these are all means by which translation is regulated, for the most part, at the initiation level. Okay. Let's see. So I'm going to describe all of this, right, using this as an example. Using this so that I can describe. It looks a little busy, but I will point out what it is that you need to know. Okay. So let's look down here first. Let's look down here first. Here's an mRNA in the cytosol. Let's say. Okay. Here's the 5' end. Here's the 3' end, and there should be a poly A tail right here. Okay. All right. So the very first thing that has to happen is that the 5' end as well as the 3' end must be recognized. Have to be recognized. How are they recognized? The 5' end, and what is recognized? What is recognized is the cap at the 5' end and the poly A tail at the 3' end. That are the two things that are recognized. What are the entities that are doing the recognizing, right? The 5' end is recognized by a dimer of EIF4E and EIF4G. And unfortunately, I will ask you to know that, the specific details of that, right? So we can't just hand wave and say, this is recognized by a regulatory protein. You have to understand, because later on it involves this. The regulatory proteins that recognize the cap is a dimer of EIF4E and EIF4G. Okay? That's a dimer. They recognize that and bind. So far, so good? So that's the cap. The the poly A tail, the 3' end, is recognized and bound by poly A binding protein. That's easy to remember. It's the green oval stuff right here. Okay? So it's recognized by, the poly A tail is recognized by multiple poly A binding proteins. Now this is important. The longer the poly A tail, the more poly A binding proteins will be binding. Right? Okay, and that's important. So far so good? The next step, okay, so cap is bound by recognition proteins, poly A tail is bound by recognition proteins, and it just happens that the EIF4E, in addition to recognizing and binding the cap, it also has an affinity for the poly A binding protein. The reverse is true. That poly A binding proteins, in addition to binding to the poly A tail, also has affinity for EIF4G. So therefore, after binding to the cap and after binding to the poly A tail, the two sets of proteins cause the RNA to fold back and interact. By doing that, then the 5' and 3' ends of the RNA will also interact. And that's a conformation that you want, that conformation right there. Regardless of how long the coding region is, the 5' and 3' ends have to interact. How do they interact? Through the binding of EIF4G and poly A binding proteins. Where do these proteins come from? Well, poly A binding protein bind to the poly A tail, and EIF4G along with the EIF4E bind to the cap. So you see the importance of the cap as well as poly A tail for initiation of translation. So far so good? Yes, ma'am.
You want us to specifically know that it's EIF4G that interacts
Yes,
with
ma'am.
poly? Okay,
That's
so
very
not
important.
EIF.
And not EIF4E. But as you can see here, even though EIF4E and EIF4G form a dimer, but it actually the EIF4E that can bind to the cap. So if EIF4G alone doesn't have a binding site for the cap. So together, by acting as a dimer, one binds to the cap and one interacts with the poly A tail. If you're missing either one, then either activity will be missing. If you don't have EIF4G, then no recognition of the cap. If you don't have EIF4G, then even if you recognize and bind to the cap, there's no, the 5' and 3' end won't come together. So both of them have to be present. Yes, ma'am.
Do those bind to the cap first before the poly A protein binds to the, or does it happen?
It doesn't matter. Okay? Yes, ma'am.
The initiation complex, is that just on, is that the whole?
Not yet. Not yet. We're about to get there, okay? When the 3' and the 5' end, and actually there are a few more EIF4, right? There's A, B, C, D, E, F, G, H. So there are a bunch of them, okay? We're dismissing all of that. We'll just say that there are a bunch more EIFs or proteins binding, and then that right there, that all of the 5', 3', all of the EIF4 and all of the proteins that bind would then recruit only the small ribosomal subunit that already has the initiator tRNA bound to it. So independently, the small ribosomal subunit, right, is bound or binds the initiator tRNA and just floating out there. And what they're looking for is this right here. Okay? Once this is formed, I say this because I don't want to repeat all of the components. Once this is formed, then the ribosomal subunit would bind to that area. Although it's drawn like lengthwise like this, small ribosomal subunit only binds if the 3' end would fold back. Okay? The whole thing, including small ribosomal subunit, including the initiator tRNA, including all these proteins here, is the pre-initiation complex. Yes, ma'am. Yes. Everything that you see here come together is the pre-initiation complex.
so bad.
Everything that you see here. So in other words, really, and I don't like this even though this showed up in your textbook, but the small ribosomal subunit and the tRNA here really need to be drawn here, right? It binds here. It only binds when the 5' and 3' end are linking or interacting with each other. Okay, so far so good? So that's a pre-initiation complex. And we're not done with the regulation of initiation yet of translation because initiation of translation still hasn't occurred. Right? Initiation of translation is what? Is when the very first amino acid is laid down. Right? You don't have that yet. So everything right now is the regulation leading to the initiation. Okay? But we, after having said that, but this whole process, a lot of people view them as the initiation because everything leads up to that, leads up to the first amino acid. Okay? Then once the small ribosomal subunit bind, or once a pre-initiation complex is formed, then the small ribosomal subunit would actually read the mRNA from the 5' end toward the 3' end, looking for, and you've heard this before, looking for the start codon, that AUG. Now that start codon, how likely is it for AUG to show up in a sequence? Extremely likely. Right? So if you do the math, it's 4 to the third. It's 1 in every 64 triplet codon. Right? It's very likely. Okay. So then it's the small ribosomal subunit is not only looking for the AUG or the start codon, but it's looking for a whole sequence that encompasses that the AUG is buried in this sequence. Okay? Just do realize that. You don't need to know what that sequence is or anything, but do realize that there may be multiple AUG. So the small ribosomal subunit looks for that. Anything that is, let's say the AUG is right here. Anything that is upstream from the AUG is not a coding region. Anything that is downstream from the AUG is the coding region. So what did we call the non-coding region at the upstream from the AUG, from the coding region? The 5' UTR. So let me give you a vocabulary word. Scanning. We say that the small ribosomal subunit, when it's reading the 5' UTR, looking for the AUG, we say that the small ribosomal subunit is scanning. It scans the 5' UTR. It scans the 5' UTR, looking for that start codon. Okay? Once it's found that start codon, then the methionine is already there. So once it found that start codon, then it will look for the next amino acid to be added, right? Then initiation, the whole process of regulation of initiation of translation and elongation picks up. Are we okay with that? And if you notice, I did not say anything about how poly-A binding proteins are released or EIF4G is released. So as the small ribosomal subunit leaves, the next small ribosomal subunit that has an initiator tRNA bound can come in. You see that? So from one mRNA, you will have repeated entry of small ribosomal subunit with initiated tRNA, and then basically you have repeated initiation. The initiation of translation process continues, meaning that the more time, the more small ribosomal subunit bind and scan, then the more polypeptide that you will make. Yes? How do you regulate the number of small ribosomal subunits that can bind? By keeping the two ends together. the longer the 5' and 3' ends are held together, the more small ribosomal subunit will enter, and then the more polypeptides will be made later on. So far, so good? Yes, ma'am.
So after that, the real initiation,
After what?
after everything.
The whole thing. Let me. I think I made it more complex by saying that. Okay. So let me retract what I said. So we'll just say that everything that I say today, up until the start codon is found, we'll just call all of that initiation, right? So whether it's called pre-initiation or the actual initiation, we'll just lump everything together, pre-initiation and initiation together. So make it simplify, just simplify things. How's that? Hopefully that helps, okay? But, you know, terminology, that's not the point. The point is that how do you regulate the number of polypeptides that are made? That's the main point. And how you do that is by keeping the 5' and a 3' and a 3' end of mRNA together linked as long as possible. Right? If you want a lot more polypeptides, then you want them to link a lot longer. If you only want to make a few polypeptides, then they don't have to be linked that long. Because as long as they're linked, then the small ribosomal subunit with the initiated tRNA can jump in and carry out scanning and then ultimately make a polypeptide. That's the point. Are we good with that? So the question is, well, how do you stop the whole thing? And we're not talking about termination of translation, right? We're not talking about that. We're talking about how do you terminate initiation? How do you stop small ribosomal subunits from entering and carrying out translation? You don't want this to go on forever and ever and ever, right? And it won't go on forever and ever and ever. So hopefully that's the next slide. No, that's oh, sorry. Everything that I said so far is in here as well. So you look for the AUG, right? So here's the same thing that we saw earlier, and imagine that the small ribosomal subunit and initiated tRNA bind to this region and then scan looking for the AUG, right? And then as long as this is kept linked, the two ends are kept linked, then you continue to have RNA, small ribosomal subunit bind. Okay, I said that about 5 different times. So, okay. At the same time, what is happening? What is happening is that, remember, what's in the cytosol? What's the function of the cap? The function of the cap is to regulate initiation of translation, also to prevent digestion by the ribonucleases, right? Prevent digestion of the RNA from the 5' end, right? Okay. Because the cap is so unique. The cap is so unique that there are enzymes that are called ribonucleases, and I think I forgot to tell you about it in this class. Did I or did I not? Okay, so there are these enzymes called ribonucleases, right, in the cytosol that will love to degrade automatically. We're not talking about degradation of bad RNA. We're talking about degradation, continuous degradation, spontaneous degradation, not spontaneous, but continuous degradation of the RNA from either 5' end or 3' end, removing one nucleotide at a time, right? So these enzymes are called ribonucleases. And what they're looking for is that normal phosphodiester bond, 3' to 5' linkage, normal, right? But remember, the cap is so unusual because it's a 5' to 5' linkage that the normal ribonuclease cannot digest the 5' end. So therefore, the cap actually literally prevent degradation at the 5' end. So far, so good? But what happens at the 3' end? That's just a normal 3' to 5' linkage. That's just a normal adenosine phosphate that is added. So the ribonuclease, at the same time that everything else is going on, ribonucleases in the cytosol will start removing the A's, shortening the poly A tail. So far so good? Yes, sir.
So this methylated one is like backwards.
Don't worry, yeah, don't write this backwards.
Recognizes it as being like 2 on both sides of the
No, it's just that just know that the ribonuclease does not recognize the cap, so therefore it's not going to do anything, is the point. Right? So it's so unique that it's not recognized by ribonuclease. Okay? So then the 3' end will get digested slowly by the ribonuclease. Okay? So think about this. The longer the poly A tail, the longer it takes for the ribonuclease to get rid of the poly A tail. The shorter the poly A tail, the quicker it takes for the ribonucleases to get rid of the poly A tail. So far so good? Yes, ma'am.
If there's ribonucleases just eating at the tail slowly, how does it even come to get to link together? Like, isn't there a competition?
I'm leading to explanation, right? I'm leading to that explanation. Okay. Right now you have the poly A tail, then there are poly A binding protein binding to poly A tail, and that's how the two ends come together. Right? The ribonuclease is not going to just remove the entire poly A tail all in one swoop. Again, it only removes the adenosine phosphate one at a time. So therefore, the longer the poly A tail, the longer it takes to remove all of the adenosine phosphate. The shorter it is, the quicker it takes. That's the answer to your question, right? Okay? So poly A tail is still there. So imagine if some of these are removed. What happened to the third poly A binding protein? It's gone. And then some more removed. That's gone. Even if you have one poly A binding protein, what happens to that poly A binding protein? It still binds to the EIF4G. What does that allow the cell to do? Still carry out initiation of translation. Once the poly A tail is completely removed, what happens to the poly A binding protein? No longer bind. What happens to the binding between poly A binding protein and EIF4G? No longer occurs. What happens to the linkage between the 5' and the 3' ends? It doesn't occur. Then what happens to the RNA, small ribosomal subunit? It doesn't enter. You see that? It's very methodical, one step at a time. I'll repeat all of that. Okay. I just want to go through that for most of you. If you just put down your pen and just listen to me, it's very methodical. It's very logical. You don't even have to memorize it. You don't have to write it down. It's extremely logical, right? It's extremely logical in that you have to have a poly A tail. Why is that? Number one, so that poly A binding protein can bind. You have to have poly A binding protein. Why is that? Because only poly A binding proteins can bind to the EIF4G. Why do you want that time binding? So that the 3' and the 5' ends of the mRNA can link. That's the ultimate goal of those proteins, right? So as long as you have the poly A tail, you will have poly A binding protein, so you will have that interaction. Now, at the same time, the poly A tail again, it's not being removed all at once. You have 250 poly adenosine phosphate, for instance, which is a really long one. Then the ribonuclease has to carry out removal of the adenosine phosphate 250 times, right? How is the in time-wise, how is the removal of 250 adenosine phosphate compared to the removal of 100 adenosine phosphate? Takes twice as long, probably, more than twice as long. Do you kind of get that? Okay? And so on a while, if it takes a while for the poly A tail to be removed completely, then the poly A binding proteins continue to be able to bind to the poly A tail, which then the 5' and 3' end continues to be able to link, which then allows for more and more and more small ribosomal subunits to bind, which then indirectly equals more polypeptides, right? And once the very last, maybe not the very last, but a few of the last adenosine phosphate had been removed, then no more poly A binding protein can bind. What does that mean in terms of binding to to the EIF4G. If you don't have a poly A binding protein, you're not going to be able to bind the EIF4G. What happens if poly A binding proteins and EIF4G won't be able to bind to each other? Then the 5' and 3' end won't link. What happens then? Then small ribosomal subunit won't bind. Some of you might think that you might be able to trick me by saying, well, can't poly A binding proteins bind to the EIF4G with or without adenosine of the poly A tail? And what is your answer?
No,
Why not?
because it needs the poly A.
Conformation. Normally, poly A protein looks slightly different, won't be able to bind to EIF4G until it binds to the poly A tail. What happens when the poly A binding protein binds to the poly A tail? It changes its conformation, right? Structure. Then the binding for the EIF4G opens up. Then it binds, right? Structure. Structure is really important. Is that pretty good? Some of you did put down your pen, so that's good. But you understand it, and that's all I need you to know is understand this stuff. Yes, sir.
If EIF4G is linked to a poly A protein at the beginning, will it go down as the ribosomal nucleus eats away at it?
Go down. Can you?
Like in this picture, it shows it all the way at the end,
I
and
see.
then
I
that
see.
one
Yes.
goes all the way.
Right.
Does it go down as it's getting eaten?
So let me. Yes. And here and I'll show you a time lapse of what happens. Okay. So we start with that. Right. There's binding. There's binding here because there's binding here. Right. When no more adenosine phosphate is there, this binding is released. That causes conformational change, thereby this binding doesn't occur. But then this binding looks pretty good for the EIF4G, so it moves down. That's how incrementally that's what happens. So very good with that question.
So does it always bind at the tail end first?
Right. And why is that? Conformation as well. So you would, and I don't know this specifically, but my guess is that's what happened, is that only the end poly A binding protein would have the correct conformation because it doesn't have another poly A tail there to affect its conformation. So this conformation is affected, but so does this conformation, right? So protein binding, whenever a protein binds to anything, that binding will almost always, 99.99999% of the time, will cause a slight conformation, no change. So that's a very good. So by you asking that, I know that you get this thing, which is good. That all is the regulation. Okay, we're gonna make it more complex. But is everybody okay so far? Everybody okay? All right. What happens to the 1% or so of RNA, mRNA that does not have a cap? That we're going to talk about that. Yes, ma'am. You have a question? Oh, okay. What happened to them? This is what happens to them. Okay. Like I think I told you that there has to be a cap. Most of them have a cap, but if the cap is absent, then there has to be some other kind of marker, right? And the other kind of marker is called IRES. It stands for internal ribosome entry sequence. You don't even have to know that. I'm just telling you that because it makes sense. So internal, which means in the middle of the RNA, ribosome entry. So this is where ribosome comes in, and it's a sequence. It's a certain sequence. Okay. So here's the deal. Absolutely, for sure, the mRNAs that do not have a cap must have an IRES. Must. You also have the case where there are a small population of mRNAs that actually have both the cap and the IRES. Okay? So again, however, no cap, you must have IRES. But some have both cap and IRES. And there's yet another subset of that where there are actually many of them, the ones that have IRES, many of them have multiple IRESs, not just one. Okay? So those are some choices that mRNAs get. So the idea is that IRES will have a similar function as the cap. Similar, not exactly similar. Yes, ma'am.
So, if the mRNA has an IRES sequence, then it won't get degraded prior to the
Right.
full
And we'll talk about how it does that.
Okay.
Right. So one of the function of the cap is to prevent degradation from pi prime end, right? But how do you prevent it without the cap? Then the ribonuclease will try to degrade it from the pi prime end, and we'll look at right now. We'll look at how that's not going to happen. Okay. So two things about the IRES. One is the fact that, and this answers Melissa's question, the fact that it has to form a secondary structure. A secondary structure is different from the secondary structure for protein, not an alpha helix or beta pleated sheet. And our secondary structure is this thing called a hairpin or stem loop, where just if you, a lot of mRNA would have complementary sequences, so the RNA just fold back and form this structure right here, which is called either a hairpin or a stem loop. Stem right here, and then here's the loop. Okay? So far so good? So all IRESs contain, would be found within a stem loop structure, number one. Number two, the start codon, AUG, is immediately downstream from IRES. So those are the two characteristics of IRES. Is one, the IRES is always found within, embedded in a secondary structure. Number two, the AUG is always found downstream, almost immediate, you know. Doesn't have to be immediate, but a few nucleotides down from the IRES. So that's relevant to initiation of translation using IRES. And then going to answer Melissa's question is, ribonucleases can only recognize and digest mRNA if it's single-stranded, right? So a hairpin or stem loop looks like a double-stranded mRNA. So therefore, it might digest this 5' end right here until here. And then once it sees that double-stranded RNA, it stops. So in fact, the double-stranded RNA actually prevents further digestion, and you don't need a cap. And actually, just the secret of the trade is that there are a lot of secondary structures in the 5' UTR. Okay, so that there, in one major functions of these secondary structures, even if you have a cap, is as a preventative measures, just in case something happens to the cap, then the 5' end will not be digested because there are a bunch of these secondary structures. Okay, so kind of a fail-safe mechanism. But again, ribonuclease will not be able to digest RNA if it were in this hairpin stem loop structure. Okay? The IRES itself is a part of the 5' UTR, right? Because the coding region, if the AUG is here, then anything upstream from that is a 5' UTR. Okay, so far so good? All right. And then more differences between the CAP and IRES is that EIF4G actually can recognize an IRES without the presence of EIF4E. Yes, ma'am.
Will you kind of just reiterate the importance of that loop at the top?
What loop at the top? This right here, the hairpin?
Yeah.
Yeah, the importance is that it prevents digestion at the 5' end, right? I guess that's it. So secondary structures, right, it can inhibit activity or can prevent ribonuclease from digesting the RNA at the 5' end. That's it. Yes, ma'am.
So when the small diagonal subunits bind, does it start at the hairpin?
We're not quite there yet. Yes, sir.
So with the absence of a cap, then there would be no EIF or E by default.
And that's what I'm about to talk about. Right. Right. So EIF4G can bind to the IRES, right, without EIF4E. So yes, so you don't need EIF4E. You don't need EIF4E, right? So EIF4G is just floating there and it sees an IRES, boom, it's going to bind. Okay? And EIF4G, once bound, it's already bound, then if there are poly A binding proteins around, then the 5' and 3' end will link. Yes, sir.
If there was no EIF4E and no IRES, would that just be digested then? Or just be damaged out?
I can see, I can kind of, may I ask you not to think in that detail because it's, all of this has, the reason for this, and I'll mention it a little bit, but it is, the reason for this is quite complex. Quite complex in that cells don't always have a nice 37 degrees temperature with pH of 7.4, right? So in inclement environment, nasty environment, that sometimes this has to be used, right? So this is only used when the cell has to, but normally this is used. So I don't. I understand that. It's just too much detail and it just might confuse you or other people more, right? So it is a fair question though, I just want to mention that. It is a good question. It's just pretty complex. Okay. So back to this. The whole idea for initiation of translation is really for the not really, but is for the 5' and 3' end to bind, right? Going back to your question then, is as long as the 5' and 3' ends interact, then yes, the small ribosomal subunit will bind, right? But once it's bound, does it have to carry out scanning? No, because the start codon is right there. So once bound, then the small ribosomal subunit can do this immediately. And again, sort of go back to what you asked. The reason why that is that if the cell has to use this system, it's an emergency. So it has no time to waste in scanning and doing all kinds of stuff. So it's an emergency. You want to carry out translation as quickly as possible. So if it's used, our small ribosomal subunit is there, it will carry out translation immediately, right? So there's no playing around. There's no scanning for the AUG and all of that. Okay. So far so good? At the same time, I will explain why scanning is important and then how that actually is kind of sort of related to IRES. But so far everybody understand the role of IRES, understand that it's an emergency and you have to do it right away, right? But why is that normally you have scanning and what does that have to do with multiple IRESs? Multiple IRESs gives the cell a choice. Scanning gives the cell a choice as well. So far so good? What is that choice? If you have multiple IRESs, then the small ribosomal subunit or EIF4G can choose any of, let's say there are 3 IRESs, and choose any of the 3 IRESs to bind, which means that the looping here can interact with any of the 3 IRES, right? If it were to interact with that IRES right there, then the 5' end would be shorter for the polypeptide. Does that make sense? And if the first IRES were used, then it's going to be a little longer, right? And shorter and longer makes a difference between having a certain domain and not having a certain domain. Therefore, the protein function is going to change slightly.
So,
Does that make sense?
yeah, can I ask a question? So if you have multiple IRESs, you're not getting multiple EIF4G?
No.
Just one IRES.
And that's a fair question, right? You hear that? So if you have multiple IRESs, the EIF4G will only choose one, will bind to one. And again, regulatory proteins and all of that, right? It's always regulatory proteins. But we'll only choose one and not multiple. Yes, that's a good question. Back then and up here, yes.
If you have multiple IRESs, is there an AUG after every single one?
Yes, yes, yes. Yes, ma'am.
Did you say that if it binds the first IRES, it makes the 5' cap shorter?
Not 5' cap,
Oh,
but it will make the amino terminus of the polypeptide shorter. You see that, right? So if it's chosen here, then all of that will not be the coding region, and all of that would have been coding for the amino terminus of the polypeptide. So the amino terminus of a polypeptide is going to be shorter. Yes, ma'am.
Would this contribute to the variability of
Yes. Yes.
proteins? Okay.
you just told the punchline. Yes. You see that? So by choosing, just by having multiple IRESs and choosing which IRES is used, you are again contributing to the variability of producing the protein. So now you have, from one gene, you would have alternative RNA splicing, and then at this level you will, alternative splicing, let's say you have 100 different variations, but then each one of those might have 2 or 3 different variations as well. Right? So you have this expansion called amplification of the variability in each step. So that's an and you just took my punchline, but that's okay. How does that relate to this? So are we saying that if there isn't a cap and if you have IRES that you have more variability than having a cap? No, that's not what I'm saying, because remember, the first question that I asked you when we talk about this is how likely is it that you have an AUG? It's pretty darn likely. So how likely is it, including the whole sequence, how likely is it that you will have multiple AUG and the sequence? And the answer is likely. So you may have 2 or 3 or even more AUGs with the initiation, within the initiation sequences and everything, right? So in scanning, the small ribosomal subunit can actually pick and choose which start codon to choose, and it will have the same effect as choosing which IRES to use.
Okay.
Right? Yes, sir.
So it could actually skip over and start codon
Right.
and choose even just
Right.
the normal.
So now we're going into even more detail, which you don't have to know, but it is interesting. This is an interesting regulation, right? So how is it that a cell can, how is it that you can have one mRNA, but yet the small ribosomal subunit will we say that it's pick and choose, but it's not really pick and choose. It's a sequencing event would lead the small ribosomal subunit to actually bind to one or the other. What do you think is, and the answer is very simple, what do you think is going on here?
Messaging?
There's always messages, but at the end, what do you think allows the ribosomal subunit to bind to one versus the other?
Regulatory protein.
Frame. Frame.
Frame.
Right?
Oh.
Frame. You might have an AUG, but if the ribosomal subunit at the beginning binds, let's say, one nucleotide upstream, so you're not seeing the AUG, you're seeing AAU, for instance, then it skips over that. So in fact, if you think about it, the 2 or 3 different AUGs and the sequences are out of frames. Because if they're in the same frame, then the small ribosomal subunit will look at all the frames. But what does that have? More contribution to variability is that in choosing the different AUG site, you're not only varying the amino terminus of the protein, but what are you also varying? Could be varying the amino acids themselves, because now they have different frames. Right? How cool is that?
Does that mean that there's an overlap in these genes?
Again, we're not talking about genes. This is RNA. There's no gene.
Yeah, but in the translation of these.
Right, but no gene. The gene is not overlap. We're talking about the sequence on the mRNA. I understand what you're saying, but don't think that way. This is not a gene. This is an mRNA, right? So in a way, yes, there's overlap, but of the mRNA, not the gene. The gene is, there's no overlap in the gene. This is the mRNA. Yes, Cam?
So for the IURS, since there's multiple of those, then how would it choose for, is it still frames? Because there's no scanning, so.
First of all, regulatory protein would help choose that, and second of all, yes, they can be out of frame as well. So yes and yes. Yes, ma'am.
So does the mRNA have the potential then to create proteins that are very different, or?
Potentially, but again, how it works is that it's not going to be that different, right? So, because if it is that different, chances are, evolutionarily, any proteins that are that different probably won't exist, won't be viable, won't be functional, right? So, there is that. Everything that I tell you, probably, on the best day, you know, on the best day in the summer where the sun is out, then this is what happens. But most of the time, you realize that everything I tell you is really of the minority, right? But the minority, even though it's a minority, but that's when you build a proper protein. And everything else is going to be destroyed. Just like RNA, right? And what I tell you probably 10% of the time, I'm just guessing, is that you will make a proper mRNA, but 90% of the mRNA will be destroyed because they're improperly made. So again, whatever I tell you is on the best day. Okay? Any more questions about this? This is huge. There will be half a dozen questions about this, okay, in different ways. I'm just guessing, I don't know. I can, I guess. Are we good though?
Yeah.
I know, I get excited because this is really interesting stuff for me. And it talks a lot about regulation and exactly, I'm trying, the reason why I talk a lot is that I'm trying to give you a view instead of just rote memory. I'm trying to get you to really think, pretend that you're an mRNA. What happens to you, right? And everything should be logical. Okay? Everything should be structural. Okay? And so I went through all of this already. So the polypeptide is going to be removed. It's just that the enzyme is called the deadenylase. Big groupy deal, right? It doesn't matter. Okay? More, as if that's not complex enough. Last thing about regulation of initiation is that all of these things that are built, for instance, ribosomal RNA and IRES and CAP, they're pretty constitutive, right? They're there already. They're there already, right? Whether or not an mRNA has a cap, it's already predetermined, or later on we'll use the term hardwiring, right? So everything is hardwired. But, however, there's some control at the external, at the extracellular level. So the cell, so when we talk about receiving a signal, what is a target cell told to do, right? The target cell can be told, so a receptor binding to the signal, the target cell, in this case, can tell the cell, presumably this is an epithelial cell in the breast epithelial cell, to produce prolactin. So what does prolactin do? Prolactin then indirectly prolong, or either prolong the degradation of the poly-A tail, or it actually directs, during the polyadenylation process, direct the cell to make a longer poly-A tail. Either way, it doesn't matter, and don't worry about it. Okay? So then an external, so everything that we talked about in terms of regulation, and on top of that, if there's an external signal that tells the cell, hey, wait a minute, you need to make even more, then the poly-A tail will resist being degraded very quickly, and that then increases the amount of casein, which is the protein that is being made. And casein, of course, is a protein a major protein in milk, right? In breast milk. And if you don't have prolactin, then that mRNA is going to be degraded a lot quicker. I mean, look at this. Look at that. That is telling, right? A lot quicker, right? So again, on top of all the regulation that we talk about now, there's an added external signaling as well. I'm going to continue. By the way, I don't know if we do, even if we have 5 minutes or 10 minutes, I'd be more than happy to have a study review, but there's so much information for this set of exam that I may not have, I'm probably going to talk all the way through at least 11:30 on Tuesday. Okay, just want to warn you that. So there might not be a very long study session, unless I talk faster. So are we okay? Okay, so if you didn't know, and you don't have to know this, but by education, I am a protein chemist. Even though I say that I'm a cancer biologist, I like proteins. And so everything I do is about protein. So this is one of my favorite things, right? Is talking about regulation of modification of polypeptide. So now that you make it polypeptide, what do you do with it? So along with the question that was asked earlier by Melissa, now you add even more variability. So you have variability in terms of transcription modification, no modification. You have variation in translation, right? Initiation of translation. Now you have not modification, but variability at the level of initiation of translation. But now you also have variability in terms of translational modification. So even if you have a polypeptide of a certain length produced, then if you have modification done, then you can have different functional proteins, or slightly different functional proteins. So even more variability. Okay? And we're going to talk about all 4 of these. I'll give you an idea of what they are, and then throughout the rest of the semester, we'll keep talking about these things, but in very specific activities. Okay? So we'll continue to talk about folding, continue to talk about covalent modification, which we already talked about, continue to talk about polymers when we talk about cytoskeletons, continue to talk about proteolytic modifications when we talk about, let's say, apoptosis. Right? Apoptosis is huge on degrading and cleaving proteins. Okay? So these are the 4 major types of modification of either a polypeptide or a protein. In this case, it's both. Some modification is done on the polypeptide as it's trying to fold, and others are done on the protein that's already folded, properly folded, but now you want to get more out of that protein. Okay? So we'll go at it one at a time. Folding. Folding itself is a modification. And this is, remember, is how we distinguish between a polypeptide and a protein, is that a protein must be properly folded. So it's not just folding. And by the way, physical biochemist, so that's like 3 disciplines crammed into one, right? So people who want to study biological processes, but only looking at molecules, so that includes chemistry, but only molecules how the molecules are made, which is more physical. So physical biochemistry, believe it or not, is an area all by its own. And so physical biochemists for a long, long time want to understand if we as human beings can take a sequence of amino acids and actually predict the structure, therefore predict the function. We can't do it yet. We can't do it yet because it's complex. So folding is not an easy thing. Folding is really, really complex, right? And the cell does it. And there are proteins called chaperone chaperone proteins or chaperonins, and there are a lot of them. There are tons of them, dozens and dozens of them, right? That are responsible for the proper folding of protein. And therefore, proper folding of proteins, right, is a translational modification. Proper folding of proteins are so important that this is indicative of it. You see this. Many chaperone proteins are, specific ones are called heat shock proteins. Where did that term come from? That term comes from the fact that if a cell is sitting there and all of a sudden a wave, the temperature increases rapidly, right, going from 37 degrees to 42 degrees. In order to keep the cell from just dying immediately, right, the cell has to shut off most of its activity and spends most of its time defending against itself against the increase in temperature. Does that make sense? So far so good? So the cell is happy and doing whatever it does, and then all of a sudden the temperature of the water increases, of the environment increases. The cell then almost immediately shuts everything off, right, and only leave the essentials. And what are these essentials? The synthesis of heat shock protein. That's how these proteins were formed, were discovered. And what are the functions of most of these heat shock proteins? Folding. That's how protein folding is, how important protein folding is, or polypeptide folding is. Okay? So I won't go on and on and on and on. So folding is important. Also membrane insertion. So a protein can be again, translation occurs only in the cytosol. Never, ever, ever on Earth would say that the translation occurs in the ER. If you learn that, quit. Drop it, forget about it. Translation is initiated and terminated in the cytosol only. Okay? And we'll talk about from the nuclear genome. Okay. However, in order for a protein, most proteins that must be membrane bound actually need to be now transported, taken into the lumen of the ER. Okay? So translation occurs in the cytosol, and then the whole polypeptide now is transported into the lumen of the ER, or the inside of the ER. Okay? To get inserted. So, folding. Let's see. Correctly folding. Oh yeah, okay. So correct folding is important. This would get destroyed, and then don't worry about the pictures. It's just these few slides depicts the fact that in the cytosol the polypeptide is made, and then it's pulled into the ER. And chaperones or chaperone proteins can be found in both the cytosol and in the lumen of the ER. Okay, and then once transported into the ER, right? Then the protein can be membrane bound.
Okay.
Did you follow the various slides there? Don't worry about the pictures, just understand that the polypeptide is pulled into the ER, and then within the lumen, this is where membrane insertion occurs, right? Very specific sequences within the membrane, within the polypeptide is going to be inserted into the membrane. Okay? These sequences, and it's not, it's indicated by the red red thing here, are called topogenic sequences. So a membrane-bound protein must have at least one topogenic sequence, or it can have multiple. So a topogenic sequence, by definition, is a sequence that signals that this protein must be embedded or inserted into the membrane. Not only does it signal that, but the topogenic sequence is inserted into the membrane. Right? So basically, the topogenic sequence tells this protein, this membrane protein that pulls it in, that that it needs to go directly into the membrane. Okay? And what do you think is a characteristic of a topogenic sequence? Non-polar.
Hey.
You knew that, right? Non-polar, right? Now, a topogenic sequence, there may be one or more. So here there are 7 of them. Okay? So we call these membrane spanning domains, or just membrane domains. Membrane spanning domains or membrane domains. And if there's one, we also say that there's one pass. So this membrane protein has one pass. If there are 7 of these topogenic sequences that also act as a membrane spanning domain, then we say that there are 7 passes. So this membrane protein has 7 passes of the membrane. So it goes pass once, pass twice, 3, 4, 5, 6, 7 times.
Okay.
Okay. So those are just the language of the trade. And what do you think these domains, how were they viewed at the level of the mRNA? Exons. Chances are, and I'm not saying that it definitely is, but let's say there are 7 exons, right? So exon number 3, exon number 5, exon number 10, exon number 13, and so on and so forth, right? That must allow for, that encode for these membrane domains that then allows for these passes. So if you have alternative RNA splicing and you splice out the last 2 exon, right, that contains the membrane spanning domain, guess what? You will not have 7 passes, you will have 5 passes. What does that do to the protein? Instead of having 7 passes, that might allow for the protein to form a nice cylinder like this. Without it, the whole protein would collapse and then prevent anything from passing through. And when we talk about transport, transport proteins and so on and so forth, passive transport versus active transport, this would be very important. You kind of have I just made a connection of everything that we talked about so far. Did you understand what I just said about exons and alternative RNA splicing and structure and okay, good. Okay. So, misfolded proteins. What happens to misfolded proteins? So proteins, even though chaperones are really important and they do the best they can to help proteins fold, but sometimes they are not folded properly. So then, if they're not folded properly, the cell doesn't want it, right? So the cell wants to destroy it. Just like mRNAs, but different from mRNA, where mRNAs can actually be bad or unwanted mRNA, or RNAs in general, can be either destroyed in the nucleus or the cytosol. Proteins, unwanted protein, can only be destroyed in the cytosol, nowhere else. Not even in the ER, where some proteins get pulled in, but not even in there. So if a protein were pulled into the ER, let's say, right? Pulled into the ER, folded, and then find out that the folding is improper, what happened is that there has to be a mechanism for that unwanted protein to be transported out into the cytosol before it can be degraded. And the reason is, the only place where you can find the machinery that degrades proteins, that degrade, not cleave or anything, completely degrade the protein, is in the cytosol. And that machinery is called a proteasome, with an A, not an O, right? Proteasome. So a proteasome is a complex of protein that carries out the digestion, again, degradation, not just cleavage, not just cutting it once or twice, but the digestion or degradation of unwanted proteins. And proteasomes are only found in the cytosol. And if you ever brave enough to take immunology, then we talk about these proteasomes at great length and how important they are within the immune response. Yes, ma'am.
If a protein gets denatured, will those heat shock proteins refold it?
No, if they're denatured, then they're cut into small amino acids, and then so they just get recycled. Heat shock, that's a very important question. Heat shock, not heat shock, but chaperonins, because not all chaperonins are heat shock proteins, not all heat shock proteins are chaperonins. So chaperone proteins are chaperonins only bind to polypeptide, unfolded. And that's a good question. So chaperone proteins only bind to unfolded protein polypeptides so that they can help in the proper folding. Once the folding occurs, right, there's nothing that the chaperone protein can do. The chaperone proteins can unfold and then refold. It doesn't do that. Okay? So there's no refolding. That's a very good question. Okay, so proteasome. Let me see where. Okay, so let's make. I think. Well, give me a second. Let me see where I am. I don't know whether I. Oh, we're good. We're good. Okay.
87.
We're good. So the last thing, and then we should have time for next Tuesday to finish up. How does a cell know? How does a cell know that this protein needs to be degraded? Right? We have an idea of why an mRNA needs to be degraded, right? If it doesn't have a cap, doesn't have a poly-A tail. How do we know what is an indication that this protein is unwanted or improperly folded? Now, improperly folded is different from unwanted because there might be proteins that are already properly folded, but maybe they've been in the cell for too long and they just need to be recycled, right? So that's why the term unwanted, which encompasses everything, right? As long as a protein is not required or needed by or wanted by the cell, it will get destroyed. So then how are these proteins, how does the cell know that these proteins needs to be destroyed? Markers, signals, right? So there are regulatory proteins that recognize that was somehow hand waving, and we know exactly what they look for, but somehow hand waving that these regulatory proteins know that this protein is unwanted. What these regulatory proteins do is they recruit an enzyme that carry out a process called that adds ubiquitin. Ubiquitin is a tiny molecule that so basically these regulatory proteins bind and a signal for enzyme to carry out modification. So ubiquitin, and the addition of ubiquitin, which is referred to as ubiquitylation or ubiquitination, it's a mouthful, right? The addition of ubiquitin. So if a protein contains not just one but multiple, so polyubiquitin, multiple ubiquitin molecules, then the ubiquitin on the protein acts as a marker, as an indication that this protein is unwanted and needs to be destroyed. Then ubiquitinated or ubiquitylated proteins then would be delivered to the proteasome and gets digested, as you can see from this cartoon, to bits, to either peptides or dipeptides or even amino acids. Yes, sir?
Polyubiquitin will only bind with a misfolded protein, is that what you said?
By an improperly folded. So if it's a polypeptide, ubiquitination would never occur because it hasn't been given a chance, right? So, and that brings up a very good general question is, these regulatory proteins, right, that bind to, let's say, transcription factors, that bind to DNA, regulatory proteins, splicing factors would bind to these sequences, the 5' and 3' splice site of mRNA. They look for a sequence. We call these linear regulatory sequences or sections, whereas the regulatory proteins that recognize protein whether or not it's unwanted is not looking for a stretch of amino acids. It's looking for a stretch of amino acid that is folded in a certain way, right? So it's looking for more of a what's the term, linear versus conformational. So it's looking for a conformational malfunction or mutation. So it's not just simply a series of amino acids, but a series of amino acids in a certain conformation. So therefore, again, that has to do with the fact that therefore, the polypeptide has to be given time to fold properly by the chaperone proteins, right, before the cell knows whether it's wanted or unwanted. That's an excellent question. One last question, sorry, and then.
It's the same way of degrading for misfolded and unwanted protein?
Yes, yes, the
Because
same.
you just wrote misfolded, so I was...
Right, so misfolded and unwanted in any way, and that's why I explained that, right? I lumped everything in under one.